today's topic is PCR, polymerase chain reaction. PCR is a technique used for amplifying DNA sequence to produce millions of copies. It is also known as in vitro method of replication. Okay, it amplifies DNA sequence to produce millions of copies. This technique is developed by scientist Gary Mullis, and he got Nobel Prize in Chemistry in 1993. There are various applications of PCR in medicines, in plant science branch, in animal science, food science. So the principle of PCR involves different events. First is denaturation, second is annealing, and third is elongation. A DNA fragment of interest is used as a template. The DNA fragment to be amplified, template of that DNA fragment is used along with the pair of primers and dNTPs and polymerase. All things are added and this mixture is kept in a machine or instrument known as thermal cycler or PCR machine. The one molecule of DNA give rise to two molecules after one round of cycle. Then after second round you get 4 copies of DNA. Third round you get 8 copies. Then in fourth round you get 16 copies and so on. The enzyme used here is Taq polymerase, DNA type of DNA polymerase which replicates DNA in vitro. Nucleotides are added. So template, primer, polymerase and 4 bases. These are the main components of PCR reaction. So here is the single DNA, double-stranded DNA template. In first stage, DNA molecule which is double-stranded, it denatures. So first it is heated, it denatures and form 2 single strands of DNA, this blue one and brown one. Then the primers, they anneal to both the strands and primers are then extended with the help of DNA polymerase. Okay, so denaturation, annealing of primers, and third is extension of primers. So from one DNA molecule you got 2 DNA copies in first cycle round. Okay, first cycle mein you get 2 copies, then in next third cycle you get 8 copies and so on. So this is basic principle of PCR. So denaturation is achieved by heating double-stranded DNA along with the other components at 95 degrees Celsius for 15 to 30 seconds. Annealing occurs at 50 to 65 degrees Celsius for 15 to 30 seconds. And then elongation at 72 degrees Celsius. That is in between 70 to 74. Polymerase elongates the primer which anneals to the denatured DNA molecule. The reaction is complete So these, for normal PCR, approximately 35 to 40 cycles are performed. The reaction is complete and the resulting amplified nucleic acid are held at 4 degrees Celsius until its analysis. So these are the concentrations of components used for PCR. Template DNA concentration is 1 picogram to 1 nanogram for viruses and short templates, and 1 nanogram to 1 microgram for genomic DNA. Primers, forward and reverse, two primers are used. Pair of primer is used, forward primer and reverse primer, at a concentration 0.1 to 0.5 micromole of each primer. Magnesium chloride 1.5 to 2 millimole for Taq polymerase. DNTPs 200 micromole of each DNTP, adenine, guanine, cytosine and thymine. And Taq polymerase. Taq polymerase is a special type of DNA polymerase obtained from extremophile Thermus aquaticus. This extremophile survives or lives at higher temperatures. So this Taq DNA polymerase extracted from Thermus aquaticus is active at higher temperatures, so used for PCR reactions. So Taq polymerase 0.5 to 2 units per 50 microliters of reaction. Instrument used is thermal cycler. Primers. Primer is a short oligonucleotide that serves as starting point of DNA synthesis. In PCR, two primers are required to bind each single-stranded DNA. These are called forward primer and reverse primer. These are designed in such a way that they are complementary to the sequence of template DNA. This you don't need designing of primers. In PCR, results are analyzed by gel electrophoresis. They are loaded onto the gel and then analyzed with the help of marker DNA. Applications of PCR in human medicines. In detecting infectious organism nowadays, for COVID, we know RT-PCR technique is used. In infectious disease detection, for several types of detections, PCR method is used. So their specific primers. Now in COVID detection, specific primers which bind to specific COVID sequence are used. PCR is also used for detecting heart diseases and cancers. Used for identification of plant species. See, for all identifications using PCRs, primers are important. Primers must be specific for that particular DNA to be identified. This you can read afterwards. Used in criminal investigation, parental testings, cloning, sequencing. In research, PCR is used for phylogenetic analysis. Okay, so this was about PCR.